right, Mr. Sister. I want you to tell me, no, better yet, stand up and tell the class. What do you want to do with your life?
I want to rock!
Achondroplasia, A meaning no, chondro meaning cartilage, and plasia meaning growth, is the most common cause of a condition called dwarfism or short stature. It occurs in about 1 in 15,000 to 40,000 newborns. You might know achondroplasia from numerous famous actors, such as Jyoti M. Gay from American Horror Story, Jason Acuna, better known as Wee Man from Jackass, and Peter Dinklage from Game of Thrones. Achondroplasia is caused by a mutation of the fibroblast growth factor receptor 3 gene, or FGFR3 gene, located on chromosome 4. This mutation causes constant suppression of endochronal bone formation. This means that the lengthening of certain bones is inhibited, causing a disproportionate short stature. We will explain this concept and normal bone growth later in this video. Besides a short stature, people with achondroplasia have other symptoms, like shortened proximal limbs, a proportionately large head, short fingers and toes with trident hands, frontal bossing, and a small midface with a flattened nasal bridge. Their average size is 131 cm for men and 124 cm for women. Factors such as intelligence, lifespan, and fertility are not affected in achondroplasia. 80% of the mutations of the FGFR3 gene are newly developed mutations, called de novo mutations. The other 20% are inherited from one of the parents. People with achondroplasia are heterozygous, which means that they have one copy of the normal gene and one copy of the mutated gene. If both parents have achondroplasia, there will be a 25% chance that the child will be homozygous for the mutated gene. Homozygous mutations cause a lethal condition. Children with this condition die, either prenatal or a few months after birth. On the other hand, there will also be a 25% chance that the child will get two healthy genes, which means he or she will not develop achondroplasia at all, even though both parents do have achondroplasia. Achondroplasia can be diagnosed through a prenatal ultrasound by determining the skull width to femur length ratio. This ratio will be higher than normal. A DNA test can be performed to confirm the diagnosis and check for homozygosity of the mutation, which would lead to a stillbirth or a very short lifespan of only a few months. To confirm the diagnosis, a skeletal survey can be used. Multiple X-rays will be made to map the whole skeleton. A skeletal survey can also detect and confirm other skeletal dysplasia, such as thanatophoric dysplasia, osteogenesis imperfecta, and achondrogenesis, in case the hypothesis of achondroplasia turns out to be wrong. So now that we have a general idea about what achondroplasia is and how it is caused, it is important for us to know how bone growth is normally organized. There are two types of bone formation: intramembranous ossification and endochondral ossification. Intramembranous ossification occurs in flat bone formation, which occurs in the ribs, scapulae, pelvis, and skull. This bone is formed directly by mesenchymal cells. Endochondral ossification occurs in the long bone formation, which concerns the extremities. This is bone formation from a cartilage model. Since achondroplasia causes shortened limbs, we can conclude that the FGFR3 gene has a more important role in endochondral ossification. We will focus on this type of bone formation and its role in bone lengthening. Endochondral ossification, like mentioned before, is a type of bone growth that occurs in the extremities. During endochondral ossification, the epiphyseal plate is created. This particular plate initiates longitudinal bone growth, which continues until the body's full size is reached. So how does longitudinal bone growth work? The bone consists out of an epiphysis and a diaphysis, separated from each other by the epiphyseal plates on both sides. These epiphyseal plates contain chondrocytes. Chondrocytes are cells which are essential for longitudinal bone growth. Bone growth occurs in the epiphyseal plate in the following way. At the outer part of the epiphyseal plate, inactive chondrocytes are activated and start to organize themselves in columns. The cells start to proliferate and push the resting cartilage outward, lengthening the diaphysis. The chondrocytes are then stimulated to mature and enlarge, also lengthening the diaphysis. Once fully matured, the cells die, and during this process calcify the cartilage matrix. Osteoclasts remove this calcified cartilage and make way for osteoblasts to build new, spongy bone trabeculae. There are many factors that influence longitudinal bone growth. Proliferation and differentiation of chondrocytes in the epiphyseal plate is regulated by endocrine factors, such as growth hormone, thyroid hormone, and sex hormones, mechanical factors, such as hydrostatic forces and muscle contraction, and locally acting secreted growth factors, such as IgG1, TNF-alpha, TNF-beta, EGF, and FGF. In achondroplasia, the FGFR3 is mutated, causing inhibitory signaling on the chondrocyte proliferation and differentiation. You can imagine that this has a big influence on longitudinal bone growth of the extremities. FGFR3 is a receptor which can act as a signal transducer for many different intracellular pathways. But before we can get into these pathways, it is essential to know more about this receptor. FGFR3 is one of the 4 receptor tyrosine kinases which mediate the responses of fibroblast growth factor, or FGF. FGFs play an important role in human development. The receptors are composed of an extracellular segment, an intramembranous segment, and an intracellular segment. The extracellular segment comprises 3 immunoglobulin-like C2 type domains, IG1, IG2, and IG3. IG2 and IG3 are involved in ligand binding. FGF only poorly binds to the receptor on its own, and therefore needs heparin sulfate proteoglycans to create a strong bond with the receptor. Simultaneous interactions with heparin sulfate with FGF and the IG2 and IG3 domain lead to sustained dimerization. Let's look at the consequences of FGF binding. So FGF binds to the FGFR, in this case FGFR3, and starts the pathway. The ultimate effect of the FGFR3 is bone growth inhibition. This is accomplished through signal transduction. Tyrosine kinase containing receptors have an intracellular kinase catalytic domain. Ligand binding causes dimerization of the extracellular domain. This brings about a change in the relative position of the transmembrane domains and in turn promote dimerization and activation of the intracellular protein kinase segment. When one kinase runs into the other, the alpha C helix moves into the right position and the activation segment moves to the outside. This makes substrate binding possible. The substrate of the first kinase is the second kinase's tail. The tail binds to the substrate site and gets phosphorylated. Then the roles are turned around and the other kinase gets phosphorylated. This process continues until both kinase segments are fully phosphorylated. The head-to-tail dimerization causes a conformational change that renders the receiver catalytically competent. The activated FGFR3 can activate multiple pathways, like the RAS-MAPK kinase pathway, the PI3K ACT pathway, the PLC-gamma PKC pathway, and the STAT1 pathway. We will focus on the RAS-MAPK kinase pathway because it regulates the growth of chondrocytes. It is also possible to interfere in this pathway for treatment opportunities, which we will explain later in this video. The RAS-MAPK kinase pathway starts with the activation of the FGFR3, which leads to phosphorylation of the docking protein fibroblast growth factor receptor substrate 2 alpha, or FRS2 alpha. FRS2 alpha recruits the membrane anchored adapter protein GRAB2, which then recruits the guanine nucleotide exchange factor SOS1. It is likely that GRAB2 and SOS1 remain associated even under non-stimulating conditions. SOS1 needs an active RAS in order to bind an inactive RAS. The recruited SOS1 activates the RAS by exchanging its GDP for a GTP. This will then activate the MAP kinase pathway. Activated RAS can bind to the protein BRAF. BRAF is an inactive kinase which is stuck in the wrong conformation. It is bound to MAC1. In this conformation, its binding site is bound to the RBDCR domain of the BRAF protein, which prevents activation by dimerization. RAS can bind the RBDCR domain of the BRAF molecule, which changes its conformation. BRAF is now able to dimerize and become catalytically competent. Once dimerized, BRAF is able to phosphorylate MAC1. MAC1 is a dual specificity kinase which can phosphorylate ERK1. ERK1 is an extracellular regulated kinase. Once phosphorylated, it can travel into the nucleus and phosphorylate transcription factors like ALK4. Transcription factors form big complexes, which create the possibility of gene transcription and therefore the formation of mRNA. The cell uses this pathway to control the cell division cycle. So RAS-MAPK kinase is responsible for the proliferation and the differentiation of chondrocytes. In achondroplasia, however, the FGFR3 receptor is mutated, which causes it to be in an active state more than usual. This is due to the fact that the mutated receptor can activate itself without the need of ligand binding. The mutated FGFR3 can cause several different types of dwarfism, varying in the severity of the phenotypic characteristics. Often these characteristics can be traced to specific locations of the mutation and the function of the segment in the protein receptor. Like briefly discussed before, the FGFR3 consists of three domains: an extracellular domain, a transmembrane domain, and an intracellular kinase domain. The most severe phenotypes are associated with missense mutations at nucleotides encoding the extracellular region, and in some cases the intracellular domain, which is something we will discuss later on. Mutations in the extracellular region cause the lethal thanatophoric dysplasia type 1. Zooming in on this region, you can see that it consists of three parts, like mentioned before. IG1 is not shown in this animation. In the extracellular domain, an amino acid substitution will cause a conformational change in the receptor. This allows the receptor to dimerize and be in an active state constitutively, despite the absence of ligand. Achondroplasia is characterized by a mutation in the transmembrane domain. If we zoom in on the transmembrane domain, you can see it looks like this. The mutation shows two transmembrane domains of a dimerized receptor. The mutation causes a substitution of a glycine for an arginine. Arginine is an electrically charged large amino acid and will therefore change the structure of the transmembrane domain, allowing the monomers of the receptor to be closer together, which will lead to an increased likelihood of spontaneous activation in the absence of a ligand. Hypochondroplasia, the mildest form of dwarfism, and thanatophoric dysplasia, which is lethal, are both associated with mutations in the intracellular domain, also called the kinase domain. A more detailed version of the domain looks like this. The inhibitory triad in blue is the most important factor in keeping the receptor in an inactive conformation in the absence of ligand. A network of hydrogen bonds between glutamate, asparagine, and lysine inhibit movement of the alpha C helix, as seen in purple, and prevent positioning of the catalytic cleft. Phosphorylation of the activation loop segment, which is seen in red, leads to the dissociation of this hydrogen bond network, thereby exposing the catalytic cleft and allowing phosphorylation of tyrosine residues. Mutations in the inhibitory triad disrupt the hydrogen bond network, and thus allow spontaneous activation of the receptor. The difference between the mutation in hypochondroplasia and thanatophoric dysplasia are the amino acid for which the triad amino acid is substituted. Hypochondroplasia substitutes a glutamate for an asparagine, thereby slightly disrupting the triad in conformation, but still allowing the hydrogen bond formation. Thanatophoric dysplasia substitutes one of the amino acids for an amino acid that cannot form hydrogen bonds, causing complete disruption and lethal disease. Another mechanism in the kinase domain involved in the conformational change can also activate the receptor. Like in thanatophoric dysplasia type 2, where the substitution of a lysine for glutamic acid in the activation loop of the kinase domain allows exposure of the catalytic cleft, even with the inhibitory triad still intact. The normal activation loop in blue changes shape as depicted in red. This causes the receptor to be constitutively active, resulting in a non-viable phenotype as seen in the disease. Thanatophoric dysplasia type 1 can also be caused by mutations in the intracellular domain. These mutations are caused by an amino acid substitution of cysteine. Cysteine has a sulfuric side chain, which, when introduced in the intracellular domain, will allow disulfide bridge formation between monomers and thus independent dimerization. Thanatophoric mutations will therefore result in a high activity of the receptor. In conclusion, different mutations in the FGFR3 can cause different phenotypes that vary in severity, one of which is achondroplasia. Even though the cause of achondroplasia is known, a cure has not been developed yet. A current treatment is limb lengthening by means of surgical intervention to increase stature. However, you only gain 5 to 10 centimeters and it requires multiple operations over 2 to 3 years. The excessive activity of FGFR3 and its signaling pathway have been the main target for developing potential therapeutic strategies over the last 5 years. The most promising treatment option is BMN-111, also called vosoritide. This is a C-natriuretic peptide analog, or CNP analog. The mechanism of action of the CNP analog is as follows. CNP and FGF signaling pathways converge at the MAP kinase pathway. CNP binds to NPRB and generates cyclic GMP, which activates different signaling mediators like CGK2. CGK2 inhibits the activation of RAF1 and consequently the activation of MAC1 and ERK1. RAF1 is part of the RAF family, the same family as BRAF. The effect of CNP on FGFR3 pathway leads to an increase in chondrocyte proliferation and differentiation, and an increase in cartilage matrix synthesis. The exact mechanism by which CGK2 inhibits RAF1 is not well established, but probably involves the activation of P38. A phase 2 study of BMN-111 is ongoing right now, but in 2015 investigators already stated that 10 out of 36 children experienced a mean increase of 50% in their annualized growth velocity. This would mean that treatment with vosoritide could be a real option in the future. So let's summarize what we learned today. Achondroplasia is a condition which leads to a shorter stature. It is caused by a mutation of the FGFR3 receptor. This receptor initiates different pathways, which leads to inhibition of proliferation and differentiation of chondrocytes. One of these pathways is the RAS-MAPK kinase pathway. This pathway can be inhibited by the use of BMN-111. Research about this treatment is still going on and might lead to the development of a treatment for achondroplasia.